Welcome to our May DataWorks Salon, managing research data across multiple data repositories. Before we get started, I'll do a little bit of background for those of you who are new to the DataWorks initiative. So FASEB DataWorks is intended to bring the biological and biomedical research communities together to advance human health through data sharing and reuse. So obviously you found our salon, but if you'd like to learn more about DataWorks, here is the website at faseb.org/DataWorks. And how do we do this? DataWorks was launched in September of 2021. One of the activities that had a quick launch was one that you're participating in right now, the DataWorks Salon. So these are monthly virtual conversations, and hopefully we will have lots of conversation today. We also have the DataWorks Prize, which is an annual cash prize recognition award, and a help desk that provides how-to guides and a chatbot to provide information on data management best practices to help you if you are new to preparing data management plans to make sure you have the resources available. And speaking of the DataWorks Prize, so earlier this year, we announced the 2023 winners of the DataWorks Prize, and we will once again highlight the efforts of the winners in a DataWorks Prize symposium. That will be on June 25th, 11 to 3 p.m. Eastern, or 11 a.m. to 3 p.m. Eastern, and this will give you an opportunity to learn about the award-winning innovations in data sharing and reuse in biomedical research. And so we'll have 7 speakers across 4 sessions. They're organized by panels. And again, you can register at our website, faseb.org/DataWorks. And so for today's salon, really excited for today's discussion. We have 3 presenters: Anna Van Gulik, the head of data review at Figshare; John Piles, the Associate Director and Principal Research Scientist at the Center for Human Neuroscience at the University of Washington; and Zefan Zhang, a researcher at the Max Planck Institute for Empirical Aesthetics. So without further ado, I will turn it over to Anna to kick us off.
Thank you very much. All right, let me screen share here. Excellent. Okay. Thank you so much for the invitation to participate in today's salon. I'm going to just sort of introduce the background here and give a little lay of the landscape before handing it over to our 2 researchers today to tell you about their experience sharing data in multiple repositories. So I'll just outline the ecosystem a little bit, and I'll also give some examples from Figshare. So this research data repository ecosystem. Many of you may be experts on the call and may be well familiar, but to make sure we're all in the same landscape here, I want to specify that there are a few different types of repositories that we'll be talking about today. One is the domain-specific repositories. These are limited to a specific type of data, specific methodology, specific subject area. Some of them are run by NIH, some funded by NIH, and others by professional organizations and research communities themselves. And the benefit of these is that they can be very specific. So you can have subject-specific or methodology-specific metadata, data standards and requirements, and that can really facilitate reuse, as well as discovery, because this may be where a research community goes to find their data sets. And then you have 2 sort of more flexible types of repositories, which would be institutional repositories, those run by academic institutions that accept many different types of data and other research outputs, depending on the specific repository, and then generalist repositories, which are, again, flexible, taking data across disciplines and across formats. And these can be used together. So there's a few different things you would keep in mind when selecting whether a repository is a good fit for your data: that discipline and format requirements, whether there are multiple different types of materials that need to be shared, perhaps software and code or images need to be shared with a data set. How can the data set be licensed? How do you intend it to be reused? if you need curation support, and whether there's any sensitive information that might require managed access or embargoes. And this is when you're considering selecting a repository. You can also consider selecting multiple repositories, either for the same data or for multiple outputs. A brief mention of the NIH data management and sharing policy that went into effect in January of 2023, because I imagine it is relevant to many of you working in biology and biomedical research. And so just a reminder that this encourages researchers to plan for data sharing in advance and to maximize appropriate data sharing, including using FAIR principles for making the research findable, accessible, interoperable, and reusable, and using established trusted repositories, including discipline-specific repositories when those are available, and generalist repositories when they are not, or institutional. And they also, NIH also lays out some characteristics to look for in repositories, such as the use of persistent identifiers, standardized metadata, provenance, and so forth. But this can be a lot to take in. So I'll highlight a couple of ways where you can look for recommendations on repositories, some resources that you can look back to later. So the NIH sharing site, data sharing site, has a list of repositories, and this includes many discipline-specific repositories, and it also points to generalist repositories here, including DataWorks, Dryad, Figshare, Open Science Framework. I also like these two resources that have sort of different search workflows, whether you're looking for different licenses or materials, or looking for a specific disciplinary repository. So one is through National Network of National Library of Medicine, NNLM, and the other one is Re3Data, which is a registry of research data repositories. Also, shout out to FASEB. I really enjoyed your finding repositories information. To help them promote these materials that they've posted online, there's a nice workflow they have, as well as a pro-con list for selecting domain-specific and generalist repositories. And I would encourage you to look at those resources as well. Because we're speaking about generalist repositories, I want to briefly mention the NIH Generalist Repository Ecosystem Initiative. This is a program funded by the NIH Office of Data Science Strategy that was launched a little over 2 years ago. And it has the goal to make it easier to share and discover research in generalist repositories, and also to break down the silos across repositories so that data can be found and tracked and reused regardless of what repository it is in, which is important when using multiple repositories, because you want those outputs to be well linked and discoverable, regardless of where they need to be housed. So the GRAY program brings together 7 different generalist repositories: DataWorks, Dryad, Figshare, Open Science Framework, Mendeley Data, Vivli, and Zenodo, working together in collaboration or co-opetition to develop standards for metadata, for having common metrics, cataloging our use cases, and doing training and outreach together to jointly support this effort. We're also helping to catalog both the similarities and differences across our repositories. So this is a comparison chart we've created that help highlight the different attributes of our repositories that may help you as a researcher or as someone supporting a researcher determine the best generalist repository for a data set. Once you've gone through the flowchart and determined a generalist repository is appropriate for part of a data set, now which of these 7 might you use? And we're currently updating that, so do look for another version 4 to be published this summer. And we've also just published a repository selection flowchart. This has fewer attributes than the comparison chart on the previous slide, but it does have a different flow to it that may work better for understanding things like sensitive data, curation, data set size, and licensing that each of the repositories offer. So now a little brief word about Figshare specifically, which will actually lead into John's talk nicely. So Figshare is a cloud-based repository for sharing, archiving, and making research of any type publicly available. We offer a few different repositories. Figshare.com is our freely available generalist repository for individuals. Figshare Plus is again open for anyone in any discipline for hosting large data sets over 20 gigabytes. And then we also offer are Figshare for institutions or custom Figshare repositories for organizations, which is an enterprise offering for a custom configured repository. We strive to offer flexibility to meet researchers' workflows by integrations, offering an API and GitHub integrations, persistent metadata like DOIs and ORCIDs, making everything open access, so CC0 or CC BY licenses are the only ones available on figshare.com to make things as open as possible, but also closed when necessary. We also offer embargoes, and to help researchers get credit for all of the different types of output of their work. And then Figshare Plus allows for larger data sets, and Figshare for Institution allows for institutional support and curation or review of those data sets before they're published, as well as some other integrations and customized tracking and showcasing of that work. So here's a few examples of organizations that are using Figshare in that way. Here are the repositories powered by Figshare for HHMI Genelia Research Campus, for NHLBI's Intramural Research Program, and then for Carnegie Mellon University, which has named their repository Kilt Hub. And that's one of the repositories that John will be talking about in a minute, where they shared a portion of their data set. As Gray, we have been creating a use case catalog across the generalist repositories. And here's a sneak peek at a new use case that we'll be publishing in June. This is the Figshare use case for how a generalist repository might support institutional data sharing with a customized repository, such as Kilt Hub at Carnegie Mellon. And this overall creates a global searchable network of those research outputs, whether they're on figshare.com or Carnegie Mellon Kilt Hub. They're all searchable in Figshare. They're all searchable on DataCite through DataCite DOIs. And it allows that enhanced flexibility to share any file type and any research output, including many that can be visualized, like these videos and images here. That every record will have a persistent identifier, including those DataCite DOIs, which are then have metadata stored with the DOI provider, and that provides for additional discoverability and metrics tracking. Support for large data sets in the institutional Figshares, and in Figshare Plus, you can have single files up to 5 terabytes in total, and use automated methods to create those data sets. And then here I'll end with an example, which is on Figshare Plus, which is an example of a data set that was shared in Figshare Plus, but also in other repositories as well. So this is the Things data collection that was conducted with NIMH researchers, as well as other collaborators, including actually, I think, at Max Planck. And so one of the features this uses in Figshare is the collections feature. So they have multiple data sets which each have their own DOI. And then the collection also has its own DOI and metadata and is searchable and citable. And this is linked to a publication and to a GitHub page. And this is a multimodal data set that was explicitly designed for reuse and includes MEG, fMRI, and behavioral responses of images, and is actually quite similar to some of the examples you'll see in a moment. And importantly, I want to highlight that this data was shared on Figshare Plus, but was also shared in Open Neuro, some portions of the raw MRI and raw MEG data sets, and then also portions of it were shared on the Open Science Framework as well. So this is an example of using multiple repositories with public Figshare repositories. And now I'm going to hand it over to John, where he'll talk about the use of Open Neuro together with an institutional Figshare repository. Thanks very much.
Okay, thank you. Let me get screen share going. Okay, so today I'm going to talk about Bold 5000 dataset. So I am a cognitive neuroscientist by training, and I do high-level vision research. and I ran the MRI Research MRI Center at Carnegie Mellon University, University of Pittsburgh, called the Bridge Center, before moving to University of Washington in 2020 to direct the Center for Human Neuroscience there. So I want to talk about today some work we did at Carnegie Mellon called the BOLD 5000 dataset. So this stands for the Brain Object Landscape Dataset. And this was one of the first kind of large-scale data sets that look to work at the intersection between computer vision and deep neural networks and a lot of the things you've been hearing about going on with artificial intelligence and cognitive neuroscience. I believe we started this work in about 2017, 2018. And the PI on this project is Professor Alyssa Amanoff, who's now at Fordham University and was a research scientist at Carnegie Mellon. And this was part of an NSF grant that Alyssa worked. Alyssa works in scene perception, high-level vision, and also the intersection between computer science and computer vision and cognitive neuroscience. So real briefly, the BOLD 5000 dataset looked to work in this intersection. So we wanted to ask questions about, can computational models help reveal more explicit mechanisms used by the brain for scene understanding? So looking at scenes, looking at images of objects, things like that. Can cognitive neuroscience help improve computer vision? So can we use data from the brain to actually help improve the models that are the basis for a lot of artificial intelligence systems currently? And then also those same features that are important for CNNs, so looking at the computer vision models, also important for the brain. So what is the intersection between brain data and computer vision data? So that was the impetus behind the BOLD 5000 dataset. So to do this, you may or may not know that computer vision models usually need a very large input space. So if you're talking about GPT-4 or something like that, you have probably at this point trillions of corpus, or definitely billions of text corpus that it's looking at. And the same thing for image models, where you're using millions and millions of images to train your data set. It is hard to get neural data from millions of images, but we wanted to get bigger than kind of the typical neuroimaging. So neuroimaging is either MRIs, we use functional MRI, which measures blood flow, or EEG, or MEG, which was mentioned in the last data set. Typically in our experiments, we're on the order of tens to maybe maximum, maybe 100 images are displayed. So the goal of BOLD 5000 was to expand this data set so that we can start to look at the intersection between computer vision and cognitive neuroscience. So we showed 4 subjects, 5,254 images over 15 scanning sessions. This is a lot for anyone who might do cognitive neuroscience or MRI research. Usually, if you're going to do an MRI experiment or an EEG experiment, you go in once for an hour, and that's your data set from one subject. So we scanned these subjects 15 times. Many thanks to our subjects for doing that. And the images we used were carefully selected by Alyssa and her team to be taken from the existing data sets that were used at the time for computer vision in building these models. So the Sun data set, the COCO data set, and the image data set. And this is unique in 3 different ways, or was unique at the time. I should say that there's a couple new data sets now, a couple that are even bigger than ours. The natural scenes data set, which is from Kendrick Kay out of University of Minnesota. But we were kind of one of the first ones. So we were a little smaller, but still pretty big. And we'd like to think, helped kind of pave the way. And then we collaborated actually on some of these newer data sets with some other researchers. So at the time, and kind of still, this is unique in a couple different ways. It's significantly larger than existing slow event related neural data sets by an order of magnitude. Slow event related is a technical MRI term, but basically means that we can kind of take the unique brain pattern from each image and look at that as a data point, which is something that takes a lot of time, but in some sense gives you kind of the cleanest brain data for a particular stimulus. Our stimulus set's very diverse. We used 5,000 images, and we chose those images again to overlap with those existing computer data sets. So this large-scale collection of data sets allows for novel training and novel exploration of benchmark computer vision data sets through neuroscience, so looking actually at the brain data. And the advantage of that scale and these sort of slow event-related designs allows for first time to kind of use that brain image data and compare that to computer vision models. I already mentioned, just real quickly, again, this is the first set of images. So 2 core values were used here from both cognitive neuroscience and computer vision in one data set. So from neuroscience, we've taken a careful step in this slow event-related design to get really good individual patterns of brain data. And then from computer vision, we took some most iconic and standard benchmark images from those data sets that have been used to train the models at the time. So there's a lot of indoor and outdoor scenes, a lot of naturalistic images, and the images are complicated and they contain object-to-object interaction, a bunch of other interesting things that result in a diverse data set of brain data. So one of the things, when we set out to build this and collect this data set was that it was intentionally designed for sharing and reuse. That was the goal. Our goal was to allow this data set to be used both by cognitive neuroscientists and computer vision and AI researchers to both investigate the brain and investigate and improve computer vision and artificial intelligence models. So I talked about, a couple of the things we did to begin with to manage and design the data set to do that. It's very large, it has this intersection, but we also tried to, as we'll get into now, which is relevant to the current talks, is leveraging the neuroimaging standards for our data collection, for processing and documentation, collecting all this data with having a CC0 license in mind that includes the data format, the data in multiple formats, and then making the data readily available in a variety of different ways. So from raw data, behavioral data, physiological data, things that I'll talk about more in a second. So what are the things we had to do to support this? So we had to do a lot of thinking about data management when we were planning this project to begin with. So our experimental design, I've talked about this slow event related things, which kind of gives unique brain patterns. And part of that design choice was to allow researchers, both in cognitive neuroscience, but in some ways, more importantly, in computer science, the ability to use the data a little bit more easily than they would if we had used more complicated designs, which would require kind of more complicated analyses to tease apart those individual patterns. So that was an idea behind our design choice, not to get just a clean set of data, but also a set of data that would be easily reusable by researchers, especially researchers across fields. We thought a lot about documentation, so we were very careful to document everything as we got started in the project and moved through the project. The data structure that we created, we were thinking about from the beginning. So how would files be organized in a way that would make sense not just to us, but to researchers who would also use this data set? The standards that we used for scanning, we tried to make those kind of standardized. We used what are called the Human Connectome Project kind of parameters with the MRI scanner, and collected support images that allow for refining and processing the data that were and are fairly standard in our field of cognitive neuroscience. And then using standards for documentation in terms of the quality of the metadata, what data is included in the metadata. And then finally, for the neuroimaging data, we use the BID standard, which I'll talk a little bit more about in a moment. So what did this all result in? So this resulted in a lot of output, which we were pretty happy with. And we chose to, and I guess the reason why I'm speaking today here, is we chose to share that data across a couple different repositories, because going across a few different repositories, and actually a few different repository types, allowed us to reach a broader audience of researchers. So we have a data set in KillTub, which is powered by Figshare, which is at Carnegie Mellon. We have a data set in OpenNeuro, which is a domain-specific repository focused on MRI, EEG, and MEG data, which requires specific data standards and is kind of the standard repository for cognitive neuroscience. We shared our images, mostly on KillTub, but a couple different other places. GitHub for the code repository, our preprint on Archive, which is open source PDF, and then finally an actual publication, which is in scientific data. So let me talk a little bit more about why we chose to do that. So again, as I mentioned, we wanted to reach different research communities, and different research communities have both different standards for how data is organized and then different use cases for the data itself. So we wanted to provide the data in different formats that were suited to those different research communities. So for example, OpenNeuro, so the OpenNeuro repository is a cognitive neuroscience domain-specific repository. And that requires, if you are going to upload data to OpenNeuro, you are required to upload that data in what's called the BIDS format. And BIDS is the brain imaging data structure. And this is actually honestly kind of a revolution in cognitive neuroscience and the sharing of cognitive neuroscience data and reuse and replicability across a number of different formats. So I've been doing this for about 25 years now. This is fairly new, only in the last about 7-8 years. I don't know what date BID started, but before that, it was a little bit of the Wild West. So we had some standards for example, in my MRI field, there was DICOM and NIFTY. These are kind of standard file formats, but we didn't have a standard for organizing that data from an experiment. So BIDS was created through a number of different collaborators, Organization for Human Brain Mapping, Montreal Neurological Institute, a bunch of different folks, but a lot of experience working with large data sets and trying to share MRI data. And the idea was, okay, we want to have a data set, a data organization that allows essentially an entire experiment to be formatted such that if I just hand you that whole experiment in BIDS format, you would know almost exactly what we did in terms of the experimental design, the parameters, how we use it, and the data would be organized such that you could actually use automated tools to either reanalyze the data in the way that we did before in the experiment or do something new. And this has been great because it finally allows me or you to go on OpenNeuro or anything in BIDS and say, hey, I know what they did in their experiment. But it also allows us to use what's called BIDS apps, which are then Docker containers of basically customized software that can just grab a data set if it's in BIDS format and do a variety of different things with it without having to do a lot of reformatting, all that kind of stuff that takes tons of time for researchers. Okay, so that's OpenNeuro. But that being said, BIDS is great for those kind of standardizations and the BIDS apps. But then if you're a computer scientist, you might not know that. That might be outside of your field. So KillTub offers flexibility to share in any format in any files. So there are things we could put in KillTub that we couldn't necessarily put in OpenNeuro. And that's why we chose to use two different repositories for sharing our data. And also, KillTub and Figshare work pretty well with a very large size the data set is. So it's 122 gigabytes in KillTub and then 560 gigabytes in OpenNeuro. So both of those repositories can handle that large size. Here's just a little bit more details about what's where. So I talked about KillTub, OpenNeuro, and then we also have some things on our website itself, so bull5000.org, and then finally GitHub, because GitHub is, again, a standard place to put code for reproducibility of code. A little bit more about what's in KillTub is already talked about what's in the OpenNeuro repository. That's like neuroimaging data. So what we did in KillTub, let's see my slides here. Yeah, in the KillTub data set, this was our data set that was targeted more on our computer science audience. So OpenNeuro is a repository targeted at cognitive neuroscience. KillTub is kind of where we steered our computer science colleagues a little bit more because we had processed the data in a way that would hopefully allow them to work with it a little bit more easily if they didn't have expertise in cognitive neuroscience. So we gave them both unprocessed raw DICOM data, just in a bundled way if they didn't want to use the BIDS format. And we also provided them some more processed data so that they could kind of take a pattern of brain data that we kind of process for them and apply that to computer vision models. So that was what we did with that KillTub data set. And again, as I mentioned before, this OpenNeuro data set, that's more for the neuroscience audience in BIDS format, again, in the CC0 license, and that really supports reuse by neuroimaging researchers. Bull5000, that's where we actually put our images. So we share our images there, our descriptions, and then we kind of use that website just as our general hub to where we direct people to figure out, okay, here are the different data formats that are available. And depending on your field, what's the best place for you to go to make use of the Bull5000 data set as a whole. And then finally code on our GitHub repository. And then a paper, because, you know, getting a paper is a good way to advertise your data set, but it's also a great way to, you know, lock in the actual methods. So we have our methods on our bioarchive paper, or sorry, archive paper. and our methods are timestamped and cataloged with our DOIs on the various different repositories. But having an actual paper where we tell you exactly what we did is usually a good way. It's also peer reviewed to share in a kind of cohesive way with the community what the data set is. Okay, so I will wrap up here and let Zafan get going. But we learned a lot from doing this. We actually had a forum about this at the Vision Sciences Society with Kendrick Kay, who I mentioned, did the natural scenes data set, which was collected at 70 and actually collected data over a year of 40,000 images. This is kind of the next step, and is now doing, there's a couple even larger data sets using video out there. So this is a new thing that is being done in cognitive neuroscience across both MRI, and EEG, and MEG, with a bunch of different data sets, again, to support the intersection between cognitive neuroscience and computer vision. But this is really hard. And one of the main things, just real briefly about challenges and benefits, one of the challenges that we found and one of kind of the lessons learned that we knew a little bit from the beginning, but we share with anyone else who might be embarking on this kind of project, is that you really want to think about this at the beginning. It's very hard to, if you want to share your data, if you want to put in these repositories, if you want to document it well, it's great to read our paper or someone else's paper and see what we did. So you can start with that as a plan. If you don't start with that as a plan, it's going to take you probably 5-10 times as much time later to figure it out. And that's both the data formatting, what data we need to collect, the metadata, so how we document it, and then the overall design of the project. So we started from the beginning, like our MRI console I designed so that it's in BIDS format, the naming when the data was actually collected. And this was 5 years ago now. So we didn't have to rename and redo all that file formatting. So that's great. And then also thinking at the beginning what repository you might want to put your data in. So we thought from the beginning, okay, we're going to go into Open Neuro. We also want to provide these resources for computer scientists. What will be a good fit for that? We decided on KiltHub/Figshare for that. And then also code repositories went in GitHub, and we decided that, well, we need a place to send people from the paper to be the hub, and that would be the website. So we kind of thought about that all ahead of time to make sure that it was in place and we could get as much impact and as much user friendliness from our data set from the beginning. And this has resulted in pretty good success. We've had a lot of people already reuse the BOLD 5000 data set for a number of different things and different studies. And hopefully we kind of set a roadmap for other repositories and projects as well. I know the natural scenes data set is now in use in many, many different studies as well. Okay, so I will finish up and I'll take questions, I think at the end. I just want to acknowledge again, Alyssa Amanoff, who started this project. This was also a grant with Michael Tarr, who's in psychology at Carnegie Mellon, and Abhinav Gupta, who's robotics at Carnegie Mellon. Ashley Marcus and Jacob Prince did a lot of the work along with me, and then Nadine Chang, who's the first author, who was at the Robotics Institute and is now at NVIDIA. Thank you so much.
Great, thank you. Zafan, it's over to you now. You all are making it seem very easy, so I'm looking forward to the discussion afterwards.
Yeah, thank you, Anna, for the beautiful presentation on the data repository, and also thank you, John, for the nice presentation, also your work on this data set, this beautiful data set. And today, what I will actually present to me as a PhD student at Max Planck Institute, to me, it seems to me that after we have the data repository, and after we have the data sets actually get deposited, the data still doesn't work in some sense. Because, for example, just one example, do you know how many papers, scientific reports published in 2016 only? I think I would like to wait 10 seconds. If anyone wants to take a guess, please respond in the chat. Just take a wild guess of how many papers are actually published in scientific reports, this single journal in Nature portfolio in 2016. I actually don't know how to read the comments, but 10 seconds.
No takers, they're scared of it.
5,000. Okay, I got one guess, 5,000. I'm have to disappoint you that it's 21,000 articles in 2016. And this number is actually also increasing over the years. So also at the same time, like this is me, the guy, and in the area of open science, everyone's sharing the data code. A lot of actually the data repository actually mandating, including, for example, scientific reports when we publish and we get accepted, the data is required to be shared. And this the problem is that the more data is shared out there, the less discoverable they will be. And this is really unfair for the data. So because in the current practice, let's say a scientist, the point that we want to share the data is for them to get reused. But for example, now I'm a scientist that I have a hypothesis on the test. The hypothesis to test neural correlative consciousness without report confound. And but the problem is, okay, now if I want to test this hypothesis, I need a data set that is actually studying consciousness. It has conditions for conscious and unconscious, and the task is no report task. Data modality should be MEG and MRI. And then as a scientist right now, what I can do? What I can do is basically brainstorm in my mind among all the papers that I know, which paper actually have the experiments actually fit these experimental conditions. And I go into this paper to check the data availability. And sometimes probably I'm lucky that I can actually find the data set. But most times the data is really not available because the data the data statements is like the data is only available upon reasonable request. But a recent paper has reviewed that's actually 95% of this kind of paper, this kind of data set is not available upon reasonable request. And so we envision the ideal practice in which a scientist has hypothesis and has the hypothesis needs specific experimental conditions and or data conditions actually to test this hypothesis, and we can just search it somewhere and directly arrive at the data set with a searching engine or something. So then here comes the big problem. When we have data repositories and we have the data source out there, and we still need a metadata that describe the data set and describe the experiment. So that's why, like I and a lot of collaborators of me that we try to work on a domain-specific metadata template. And so we start from one metadata template in human cognitive neuroscience from the colleague of me, and we have some hybrid workshop to actually work on this huge metadata template. At the same time, we get a lot of offline feedback from colleagues in my research institute, and we also get some online feedback from our advisory board, and we come to our current version of the domain-specific metadata template in human cognitive neuroscience. So our metadata template, it is notes that we build this domain-specific metadata template using CDAR, and the major advantage of this platform is that when we build the repository, for example, the data modality, we can actually, the options could be supported by existing ontologies, and is linked to the existing ontologies database out there, for example, BioPortal. So that means the whole metadata template is, the vocabulary is controlled. And so this is the overall view of the metadata template that we have we put out together as for human cognitive neuroscience datasets. So first we have the section for dataset, including what the pre-processing information, what is the data standard, and if it's high-level datasets, what is the source dataset, and also the crediting acknowledged information, for example, who is contributor and what is related project of this dataset, and also the experimental information in which this dataset was collected. For example, the ethical proof information, experimental paradigm, and the subject information, and also analysis. And so the datasets, in the dataset section, we ask, for example, what is data modality and what is data acquisition technique? The data modality here refers to, for example, is the behavioral data, physiological data, or neuroimaging data. And also if it's neuroimaging data, we ask what is data acquisition technique, for example, MEG, MRI, or ECOG. And also we asked what is a pre-processing, if there's a pre-processing pipeline, which pre-processing pipeline is actually applied to this data set, and what is data standard, like John previously mentioned, for example, BITS is also one of the data standard that's available for participants, for the users of this of this template to choose. And also what is the source data set if it's a high-level data. And also we have the experimental section. Basically, we ask what is the area of the research, whether it's clinical trial what is the protocol and where is the pre-registration of this experiment. And we ask about ethical approval information, like when what is the dates that the ethical approval enables this data to be reusable. And also the experimental paradigm, for example, what is the experimental paradigm that is actually applied to collect the data set, and what is instruction in the experiment, throughout the experiment to the participants, what is sensory modalities, visual or auditory stimulus type response, how the participant makes response, what is the area of interest in the neural anatomy, and what's the corresponding publication. And also we collect the subject's information, the participants, which features, which attributes of the participant actually collected, the demographic, medical, occupational, psychological. And also we also going to, because this metadata template is not only for metadata template to describe the data itself or experiment, also the experimental code in case the code is also attached to the data set itself. And also the accrediting and the acknowledgement section, we ask who is a contributor to this data set and what is their information and how could people actually contact these these contributors after it's published. And so, and this is an overview of the metadata template. So I just want to show you that with this metadata template, we are able to describe a data set, for example, with MEG and behavioral data sets defined by data acquisition technique in base format defined by data standard of 30 participants, number of participants with schizophrenia, attributes measure of participants, medical, who are instructed to detect and discriminate, which is instruction, visual, which is sensory modality, face, which is a stimulus type, stimuli that is background mask, which is the experimental paradigm, and respond with the button press, that is the response. So this description, all of them this description is completely described in controlled vocabulary supported by existing ontology out there. And so who cares about our metadata templates? Very fortunately, we have so far managed to integrate our metadata template with OSF and Dryad. And for example, on OSF, it's been, you can already find our metadata template, it's called Human Cognitive Neuroscience Data. And when you open the OSF landing page, and you can go to the metadata section, and you will find at the corner, there's a section, there's an opportunity for you to select the metadata template to describe this project and the data set that you are proposing. And also, we are also working on right now the integration with Dryad, and I think it's also available to the public very soon. So as again, we develop the metadata template. We envision the future that future researchers can actually get from the hypothesis to the experimental, to the restrictions of the data sets that they have in their mind to test the hypothesis, and they can come to the data set itself directly. And I call it a data highway. And this, and also thanks to my scientific and open science collaborators on this project.
Great, thank you so much. I would say all three of our speakers provided a wealth of information and background. Certainly, I have questions. That's supposed to be my job, right? But you know, if you in the audience have questions, feel free to add them in the chat. You can also feel free to raise your virtual hand, or you can just unmute and ask your question. I will start off with one that was just resonating as all of you spoke. I mean, I joked about it in the transition between John and Stefan, that you make it sound so easy. And you know, there was obviously a lot of planning. You both alluded to that in your presentations. You know, what would you say is the biggest hurdle for research teams to get over when planning? And this touches on a question that was in the chat. Do you do a pilot of how you organize your data before you go to, say, 40,000 samples?
I guess I'll start with that. So the answer is yes. I mean, definitely good practice in cognitive neuroscience is to always pilot your experiments, because an MRI experiment is $650 an hour, usually something like that. So we're always going to make sure that we're doing the right thing before we move forward. But yes, we tested our data pipeline in this case, and I believe our colleagues with these other data sets I mentioned to kind of do that same practice. Definitely with bids, so getting the data off the scanner, imported, formatted into bids, and making sure that that all kind of worked. Yes, I believe that we tested that out. And then we talked to Anna at the time, because she was also at Carnegie Mellon, and looked at the landscape to make sure that we had the correct metadata that we would need and weren't missing anything. The idea is, again, being there that if we didn't do something at the beginning, it was going to be much, much harder to do it later. So we wanted to make sure we had all the needed data, and we thought carefully about what our colleagues might want to use. So another example of what we did in this data set, and what I've done in another data set I collected, which will be publicly released at some point, is to think about processing pipelines. So how a neuroimager might reuse the data. And sometimes we pick one method versus another method. We actually collected the data to allow multiple methods to be used, with the idea being reuse. So that, okay, one group does field map correction, is the example I'm thinking of, this way, another group does it that way. We collected the data that would allow them to do it both ways to facilitate the data use. And we tested that at the beginning to make sure it all worked. So yes, that is a very good idea.
Great, thank you. Other comments, Stefan?
Yeah, yes, and also as because I'm based in Germany right now in Frankfurt, and in every research project, when we involves collecting data, especially contains personally identifiable information of the participants, we have the big issue with the GDPR. The GDPR limits what kind of data set actually you can share or you cannot. So then basically you must already consider whether you will share your data, how you will share your data before the experiment started, because you have to make sure that you get participant consent, and you have to make sure that the data sharing information is also in the consent form. And also you have to make sure that this is also approved by the ethics committee. And at the same time, as you have to make sure that sharing this data set is GDPR compliant. So I think sharing data sets is particularly sensitive in Europe, and we must think about it beforehand. And yes.
Yeah, that's a really excellent point. And GDPR is a little bit more complicated than it is in the US and other places, but everywhere, man, I was on the board of the IRB at Carnegie Mellon for 5 years. So everywhere, that data sharing does need to be in your consent if you're collecting data from human subjects. So you absolutely, I mean, Stefan is so right that you really need to think about that ahead of time, because if you decide you wanted to share the data later in a public way, the only way to do that, if you didn't think about it before, is to try to go back and track down every single subject and get their consent. And that's something you really don't want to do. So that absolutely needs to be thought about ahead of time.
I really appreciate you lifting that, because you know what it came through in all the presentations was just planning ahead and thinking about how your data are going to be reused. Whereas I think, and I'm going to gather that all three of you would agree that this is the big culture change, and it's something that we think about with the DataWorks initiative. Instead of having data repositories be an afterthought of, oh, I'm already done, it's thinking about how you are going to you know, what are the potential reuses of your data, either by you and your research team or other research teams. So, you know, if there is, and you've given so much advice, all of you, throughout this, but is there one area that you have found through your own experiences that there's always that gap, that it's underplanned for, that researchers should really put more time into it?
I guess I would say, I mean, the planning ahead is, again, I can't emphasize that enough as to how much harder this is if you don't plan ahead and you choose to do it later. So, but also to try to plan for the unexpected, documenting your experiment very carefully, and this should be the case in all science, but doing it in a rigorous way. If there is a standard in your field about how to document that data, in other like wet neuroscience fields, or we have a lot of biology folks here using tools for like online lab notebooks, for example. And for us, it would be keeping careful scan logs, so that if there is something that we didn't think about, that hopefully later that we have that data in a format that we can get to pretty easily. So being just organized about all the information you record and to record as much information as possible as you collect your data is really important for unanticipated things that someone might ask for later. I don't know if Stefan has other ideas there.
Yeah, I agree with everything you said. I think you have said everything I want to say.
Yeah, data management for data sharing, right, is the planning part, and maybe to go with that, also planning on the time and budget that's required for those activities. If you're thinking of this in terms of a funding model, data management sharing plans and budgets, and also just thinking of this as a part of the research process that is important, not a secondary task, but really a primary part of the research being conducted and of the scholarly publication of the work that you have staff time devoted to doing that documentation throughout and preparing the data set. That's probably something we run into in the repository side the most, is like, it's an emergency, the paper, the publisher needs the DOI tomorrow morning. We don't have any time to document it. We just need the data set DOI right now. And oh yeah, we didn't put metadata in either. So thinking about that in terms of dedicating effort to it. And hopefully that will be rewarded effort by funders and institutions and professional research communities as well.
Yeah, I will. That's a really good point, and I think it's something to definitely emphasize. And there are costs there. You know, there are absolutely costs in staff time, and as I keep saying, planning ahead will save you that staff time. But even then, there's still costs in person hours of doing this. But also looking at the landscape of repositories, what is free, like OpenNeuro currently is free. We had Kiltub Figshare, which was provided by our institution. But if we didn't have that and we had to do Figshare Plus or another option for a paid repository, that would have been budgeted into the project and the grant and the funding in some way. And it would have been big trouble if we hadn't, again, thought about that ahead of time.
You consulted your librarian ahead of time on those resources. So use those institutional resources.
Yes, and if you don't have the institutional resources, my colleague Stephanie dropped in the help desk guide from DataWorks about budgeting. I'm glad you lifted that, Anna, because that is something that the planning and the budgeting is something that I know the NIH community is learning about with the newish policy. I apologize. You were going to add something.
Oh, good. Todd.
So I just want to like about this course actually, because I'm a PhD student right now. I'm doing a lot of scientific bench work, and I'm involved in this field flow project. And this project, my colleagues actually have the idea of ask me to track the cost of my time in my open science practices. And for example, I track the time. For example, I spend on doing the registration. I spend on sharing the data, spend on sharing the code. And actually right now, my data, my after basically 2 years of this practice, I find that actually in general, sharing data and sharing code is costing basically 3 to 5% of my total working hours. So yeah, just a quantitative number to present to you guys how much time that actually costs for us actually on data sharing and the code sharing.
That's really cool. Actually, that's super interesting. I love that idea.
Yeah, that's cool.
One other thing I do want to say, and since we have hopefully a lot of researchers here and PIs, is that my field and biological sciences, probably physics, in some ways has not really, I feel like, recognized how much cloud data storage costs. So before you have sticker shock, you might want to check, and then also realize that institutional repositories or paid repositories, whatever, just if you do it yourself, it's really expensive, and that's why this is so much. If you look, so I manage MRI Center with terabytes of data that gets stored and moved and processed. And I pay at this point Amazon and or Google Cloud to do that. And I would encourage all researchers, just if you are collecting large data sets, to go look at the storage and transfer costs for all the cloud services, Azure, Amazon, Google, and understand how expensive that is, and not to probably kind of get in front of the sticker shock you might have, either in paying for that yourself through your lab, or your institution paying it through an institutional repository or something like that. That this is not cheap or free. These companies have kind of led us to believe that by giving us a lot of free stuff, but it is not free. And just want to kind of throw that out there, because I've talked to lots of colleagues who've been surprised if they have to foot the bill themselves. But it's so important for open science, so that's kind of a thing we have to deal with as a community.
Thank you for lifting that. And I recognize, like, we're at the top of the hour, so I just want to take the opportunity to thank our speakers. This was really great. The feedback in the chat has been much gratitude for everything you shared in your presentations, also in the discussion. You're getting some applause too. But you know, and I'm grateful that I learned a lot. So I appreciate today's discussion, and then going into the costs. These are all important things for researchers to think about, not only just for compliance, but thinking about how you're going to reuse it. You're planning data management to plan for reuse. I love that. Just a reminder that our next Salon will actually be the DataWorks Prize Symposium. And please do register for that. That should be a great extended celebration of our 7 winners. And then we will resume the regular Salon series after we take a little summer holiday break in September. But thank you again to all of you for joining us today.